newsletter just with monthly updates about what's going on at the lab. If you would like to stay engaged, just sign on up. Okay, pass that around. Thank you.
All right, thank you. Thank you all for coming. My name is Jason Wang. I just finished my third year as a PhD student at USC, and I am in Professor Donald Manahan's lab. So we study mostly physiology of marine invertebrates. And I'm going to talk to you today about how we kind of apply some of our research into informing sustainable aquaculture. But before I get too much into that, I'll just talk a little bit about how I got here to where I am today and why I'm interested in the things that I'm interested in. So going way back, so I grew up in North Carolina, and Father's Day is next week. So here's a picture of my dad and me taking me to the beach, and each summer we would just go fishing, dig up crabs, look for little critters. So I was always really interested in just finding animals, interacting with nature, and then that's how I kind of decided to study biology. And so in high school, I had a really good AP biology teacher who really instilled these principles in me of not just appreciating nature, but understanding how it works and what drives life to function the way that it does. And so in college, I had a great opportunity to spend a semester and a summer at Duke's Marine Lab. And it's pictured over here in Pivers Island in North Carolina. And so what's great about this marine lab here is that like much of the East Coast marine labs, they're located on estuaries, which is basically where the river meets the ocean. And so from an environmental perspective, an estuary can be a very stressful environment. You have the fresh water from the river coming in, meeting the salty water of the ocean, and then all of these temperatures, salinity, different variables can fluctuate throughout the day, and depending on the tides as well. And so this is where we began to start asking questions like, well, there's obviously an abundance of life in these ecosystems, so how is it that they're surviving in such changing and diverse environments? And so, for example, we have this mud crab here who can breathe not only in water, but also in air. So in this intertidal system, there's specific adaptations that different animals have and are able to utilize in order to survive in what to us may seem like a very stressful environment. And so this is kind of where I began to value being able to bring animals from the environment into the lab to begin asking them questions of how they function and what is it about their biology that allows them to survive. And so I went on to do my master's in the other Carolina, in College of Charleston in South Carolina. And this is where I began to take experimental physiology a little more seriously. So taking those same questions of bringing animals into the lab and then just probing them more to ask what is it specifically about the molecules inside the body up to the physiology that allows for different adaptations to persist. And so this is kind of that lab's claim to fame is they kind of put a shrimp on a treadmill, which is, it seems ridiculous to some people, but it is important because if you want to understand and ask the question to the shrimp, how is it that you're able to breathe in such low oxygen environments, you have to have some way to challenge them physically and also to kind of elicit the physiological response that you want to study. And so just being able to come up with different clever techniques to kind of ask different questions about the animal's physiology is kind of what got me excited about experimental physiology. And so what I had learned from those experiences was that what seems like a harsh environment to us isn't necessarily that harsh to an animal. There's a huge plethora of diversity of life, and all different animals have different adaptations so that what seems like a stressful place to us is actually another animal's home. And so each animal has a unique physiological strategy, and then by studying these physiological strategies, we can actually then try to figure out how nature does it best and how we can use that to our own advantage. And so that brings me here today to this beautiful facility at Wrigley. And this is a really great environment because we're surrounded by the natural MPA. So the waters are very pristine. We're able to utilize that water for our culturing needs, as well as collaborate with a lot of different labs. So there's labs all over this campus that I hope you'll be able to see. But we have people studying everything from genetics to sediments to geology, all the way up to fish and other marine animals. So there's a lot of great collaborations going on here, and it's a great place to work for those reasons. And so that's kind of why I became interested in studying marine biology. But why should anyone else care about marine biology? And so the first reason I'll give is just for environmental monitoring and sustainability. So this video here is actually showing a tank full of oysters and algae. And you can see that over just the course of a few hours, all of this algae is going to get depleted by the oysters. So these oysters are actually filtering water at very high volumes and very high rates in order to clear out what seems like a very dirty environment to us. So they provide a very good ecosystem service, and you can also see there's a lot of fish and things scurrying around. Well, it's hard to see in that video, but they provide a very good 3D reef structure, and they also provide a lot of different habitats for other animals to utilize. So other than the environment, marine organisms are also responsible for a lot of the biogeochemical cycling that happens. So this is moving up to a much larger scale. This actually shows a satellite image of a region of the ocean. And all of these plumes are actually algae that are able to photosynthesize. And so you can actually see these huge masses from space. And what's happening is that they're actually turning the CO2 that we emit into sugar and fixed carbons. And then what's even better is that when they're done with this process, they sink down to the ocean and carry all of that fixed organic matter with them. So they're a really important pump in our ecosystem that's able to sequester a lot of carbon for us. And so another reason why we study marine animals is because they're actually very relevant to physiology and medicine. So you might be wondering, well, what do I have in common with a sea urchin, for example? Well, it's actually because of the sea urchin that we know a lot about cancer treatments, because the regulation of the cell cycle was actually characterized in sea urchins. And the reason for that is that sea urchins, much like us, they go through their first cell divisions in the exact same way. And so they're very consistent with this and easy to study. And so by studying a lot of them, we actually figured out what proteins are responsible for regulating the cell cycle. And then if you think about what happens when the cell cycle isn't regulated, that's when you get uncontrollable cell growth, which is cancer. So by studying marine animals, we actually learn a lot about ourselves as well. And then finally, we also develop a lot of different biotechnologies for marine animals. This is actually a jellyfish that's able to form a green fluorescent protein. And so by extracting this protein and making it on our own, we're actually able to start labeling a lot of other proteins, which we can then study how they interact in the body. So there's a lot of different uses for marine animals. And the one I'm going to be talking about today is for food security as well. And so kind of like how civilization was boosted by the formation of agriculture, our population has been growing on an exponential curve. And so we're going to need more and more food in order to sustain that population. And so this graph from the FAO actually shows that a few decades ago, our capture fisheries production actually started to plateau around 90 million tons per year. However, we started growing more food more efficiently from the ocean, and our aquaculture production is actually still increasing in order to maintain that rise of food demand from our global population. And so food security is going to become a very important issue in the next few years. And so being able to do it in the right way and do it in a sustainable way is going to become very important. So there's a lot of different challenges that aquaculture practices face. So depending on the species that you want to grow, they all have different advantages, disadvantages, and they also have different challenges as well. If you take this hypothetical fish tank and you stock it with a very high density of fish, there's a lot of different things that have to go in. There's going to be food, there's going to be antibiotics, different things like that. And then furthermore, with all the crowding, a lot of waste is produced and deposited into the ocean floor. So not all practices are very good for the environment. While they may produce a lot of food for us, there's a lot of different just challenges that have to be faced. In addition to environmental impacts, there's also spatial impacts as well. These shrimp farms, you can see they dramatically alter the coastline. And then every time they harvest this, they have to drain out the water, bring out the shrimp, and then a lot of that water ends up back into the ecosystem. So figuring out how to grow food, not only quickly, but also sustainably is going to be a big challenge in the future. So we kind of believe that the Pacific oyster is actually a very good sustainable model for growing seafood. And already over 500,000 tons are produced each year. And the reason they're a good sustainable model is you don't have to input that much food. And in fact, if you just put them out in the ocean, they're actually filtering a lot of the algae from the ocean anyways, just like that video I showed before. And so they're actually cleaning the water, and they're also providing a lot of 3D structure or reef-like habitat. However, the oyster also has its own challenges, and that's seed production. Kind of just like how a farmer would have to choose which seeds to invest in. The oysters actually have a larval form, which is very vulnerable, and that's kind of the life stage that needs to be addressed right now in this sustainable practice. And so much like a butterfly or another insect might have like a caterpillar larval form, most marine animals actually have a larval form as well. So we think of an oyster as kind of like a rock-like, non-moving creature, but as babies, they actually spawn into these little tiny oysters that are swimming freely inside the ocean. And so each female can actually produce 50 to 200 million eggs per spawn. And then the larvae actually spend a few weeks to a few months in the water column where they're susceptible to predation, they have to find food, and they have to be able to grow very quickly. And so in the wild, making it to adulthood is sort of analogous to winning the lottery because there's so many things that can go wrong. And so when we think about how this low survival translates into aquaculture, we want to know how should an oyster choose which larval seeds to invest in. And so what makes a good seed in the first place? Well, first of all, we want one that has low cost and is able to grow quickly and is resilient and hardy to environmental change. And so if you think about this life cycle, during the 2 weeks to several months that the larva might fend in the water column, it has to be able to fend for itself and grow very quickly in order to reach a size where it can settle and metamorphose. And so in terms of the industry, a low cost might include something that requires less food. So this is showing the algal food that's being grown for the oyster. So something that is able to turn the least amount of food into the fastest amount of growth is going to do a lot better than something that takes a lot of resources to grow the same rate. And so these are actually different genetic families from either family 6 or family 7. And when crossed together, we can see there's already a very strong genetic effect of whether you feed the same oysters from the different families the same amount of food, they may grow at much higher or slower rates. And then finally, if you add on to that environmental change, these are actually baby oysters that are exposed to future climate scenarios. And we can see that the low pH that's predicted in the future actually has a very negative effect on their shell formation. So finding resilient strains of oysters that are able to grow in the light of the future environmental change is very important. And so behind all of these processes are actually a whole suite of measurements that we have to know how to ask the animal. So we study everything from the molecular biology and genetics of the animal to their biochemistry and protein metabolism, all the way to their physiology and how that affects the organism as a whole. And so how do we study animals that are so small? So this is actually, just for scale, a piece of hair and then a 2-day-old oyster. And you can see that this oyster is actually even smaller than the width of my hair. And so you can't just ask, just like you can't ask a shrimp, how are you breathing underwater in such low oxygen? If you're studying something so small, it's even harder because, they're so small. So, add on top of that, they all have their different complex behaviors. They're constantly, swimming around or not swimming. They can be feeding, not feeding. They can be doing all sorts of different things. So how do we get rid of this variation when we want to study them? And the answer for that is to study millions and millions of them. And so upstairs, we have a huge culturing facility where we're able to grow millions of larvae. This tank actually shows each tank is 200 liters of water, and we can fit in about 4 million eggs per tank. And so if you want to ask a larva, how fast are you growing? We find it's actually best to ask millions of larvae, and then they may each give us different answers, but on average, they're going to give us a very accurate and precise growth rate. On the other hand, if you ask everyone from my family how fast we grow, or if you ask someone from an NBA player's family how fast they grow, you might have very different answers. So we want to parse out that variation that's due to genetic background as well. And so we actually have the capacity to grow genetic lines of oysters in order to see which families do better and which families grow faster, and ultimately which families are going to be better for sustainable aquaculture. And just as a side note, when the oysters aren't in season, we also work with sea urchins like we are working with right now. And these are actually a lot of fun to work with as well. So this is a video of a spawning sea urchin. And when we inject them with potassium chloride, they actually release their eggs for us. And so different species have different amounts of egg quality and egg numbers as well. So by comparing different species, we can also begin to learn what strategies allow different larvae to grow faster or not grow faster. And so what do we do with all these larvae anyway? So we subject them to a lot of different physiological experiments. And so, for example, this picture here is showing a feeding rate assay. And each little speck is actually one sea urchin larva. And so by putting these sea urchin larvae in known amounts into little cylinders with known amounts of food, we can actually measure how quickly they're feeding. And then we can also put them in these sealed containers where we can measure the amount of oxygen over time and figure out how fast they're breathing. And so taking all these things together, we know how much energy they're consuming, how much energy they're burning, and then we can put that all together to figure out what's going on inside of their body. And so the goal of all this research is to kind of build this budget for growing a larva. So what does it take for one oyster to grow? And so if you just follow along this diagram with me, there's an algal food, which might be composed of proteins, lipids, or carbohydrates. And then I'll just focus on protein for now. But that protein is taken up by the larva. It's digested and broken down into its amino acid building blocks. And then those amino acids are then synthesized into proteins that the animal can use. Now, some of these proteins will end up being deposited and kept as growth. But then what we're finding is that actually a lot of this protein, up to 80% of it, is actually being degraded every day and turned over. And we think this is because of the different purposes that proteins serve in the body. And then all of that is to say that growth is a very inefficient process. So just like all of life, not just biology, you can't avoid paying your taxes. And so each time you consume an energy building block, you have to waste a little bit of it. And in this case, we're actually wasting a lot of it. So figuring out which animals are able to avoid paying these taxes the most and able to deposit the most growth is actually what we're interested in now. And so just to summarize, the sustainable shellfish aquaculture has a very vulnerable larval stage. Of the millions of eggs that are produced by oysters, only a handful of them actually survive to adulthood. And so a lot of that is because the cost of living is quite high, and the larvae have to deal with all sorts of environmental changes. And so being able to avoid the high cost of living and being able to grow fast in a different environment is very advantageous. And so we're studying more of the metabolism that will help us predict which winners and losers will be successful in different environments in the future. So that, I'd like to thank everyone at the Wrigley Marine Science Center for their support and for the facilities being used. And then also everyone in my lab, my Professor Donald Manahan, as well as the other postdocs and the other graduate students. And I'd be happy to take any questions.
Over in Cat Harbor, I thought they were raising some sea bass hens. Does your facility have anything to do with that?
We're not directly linked to that, but we actually used to keep some of our oysters out in Cat Harbor as well. But now we're starting to move into keeping our oysters in the Blue House facility over here.
Okay.
But, yeah. Yes.
When you mentioned your genetic studies of oysters, are you using any of the genomic technologies like NGS or genotyping, or you're just growing sort of families that you would select traits from?
Yeah, so we work very closely with Dennis Hedgecock, who's a very famous geneticist at USC as well, and he's been managing these pedigree lines using a lot of very precise genotyping technologies.
And are you able to infer any of the phenotypes that you see in oysters to genetic information that you get from them?
Yeah, so again, from the Hedgecock lab, they had a student who was able to, they did a lot of work with growth heterosis, so hybrid families actually grow much faster. So by knowing the genotypes and then producing, thousands of, I guess, the next generation of oysters, they actually were able to genotype the next generation and connect that to the faster growth rates and identify several regions of the genome that are correlated with faster growth. So there's still going to be a lot of studies that figure out what's in those regions of genome, so how they actually link to physiological processes.
Anything else? No? Okay, well, thank you so much.
Thanks, Jason.